The overall goal of this procedure is to use flow cytometry to isolate the stromal vascular fraction from human adipose tissue for phenotyping of the different immune cell populations found within the adipose tissue.
This method can help answer key questions in metabolic research, such as which immune cells accumulate in the adipose tissue of obese individuals and to which extent. The main advantage of this technique is that it allows for the simultaneous measurement of multiple markers on immune cells and the identification and quantification of these immune cells in adipose tissue. Although this technique can provide crucial insight into adipose tissue inflammation, it can also be applied to other organs and tissues. Generally, individuals new to this technique will mainly struggle with stromal vascular cell contamination and loss of cells during the staining procedure. Two of my PhD candidates, Suzan Wetzels and Bijnen, will be executing the protocol.
Begin by using a scalpel to mince 1 gram of adipose tissue biopsy into approximately 2 mm squared pieces. Transfer the pieces into a 50 milliliter centrifuge tube and add 10 milliliters of collagenase solution to the samples. Incubate the tissue fragments for 60 minutes in a 37 degrees Celsius water bath at 60 cycles per minute, followed by filtration through a 200 micrometer strainer into a new 50 milliliter conical tube. Rinse the filter with 7 milliliters of PBS before centrifugation. Then use a pipette to remove the supernatant without submerging the entire pipette tip into the solution.